What are genes?
Genes are the basic unit of heredity. A gene is a sequence of nucleotides containing information on specific traits that shape the characteristics of an organism. Each person actually has 2 copies of each gene, one comes from each parent. There's not really a typical size for a gene. Since the number of base pairs in a gene can range from just a couple hundred base pairs to millions of base pairs. Most of the genetic information in each of our genes is actually the same from person to person. In fact, differences in less than 1% of the DNA in our genes accounts for all of the physical differences between people.
What are alternative solvents?
Alternative solvents are relatively environmentally benign solvents that can be substituted for the more hazardous choices, which, while they may be traditionally used, may have established precedents without environmental safety or toxicity concerns in mind. One such example is 2-methyl tetrahydrofuran. As a solvent, it possesses similar characteristics to the widely used dichloromethane and tetrahydrofuran solvents, but with significant environmental advantages in that it is produced from renewable feedstocks, like corn cobs and bagasse, and is easier to separate and clean up. You can compare the chemical structures of 2-methyl tetrahydrofuran. Left, with dichloromethane, center, and tetrahydrofuran, right, below.
What does brining do to meat?
Soaking meat in a salt solution, the definition of brining, helps separate the long filaments that make up muscles, myofibril, by dissolving the proteins on their surfaces. With enough salt, the actual filaments start to break down, and both effects make the meat seem more tender. Additionally, salt allows proteins to hold onto more water, which helps prevent your steak from drying out.
What is a nucleobase?
Nitrogen-containing molecules that form nucleic acids, adenine, cytosine, guanine, thymine. Uracil are the major nucleobases, form base pairs via hydrogen bonding between the 2 helical strands of DNA.
What is an example of an alternative green reagent?
In a similar spirit to alternative solvents, alternative reagents are relatively environmentally benign reagents that are used to replace more toxic ones. One example of an alternative green reagent is dimethyl carbonate, which can be used to effect methylation and carbonylation reactions. Traditionally phosgene or methyl iodide have been used to carry out this same reaction. But the drawback is that these reagents are significantly more toxic and are thus also more costly to dispose of properly. Dimethyl carbonate is a non-toxic compound and it can be readily produced via an oxidative reaction of methanol with oxygen, thus avoiding any environmentally hazardous synthetic procedures.
Is it better to use chemical-free products?
There are no such things. Everything is made up of chemicals. Be careful when choosing which ones you eat or use, but the truth is, they're all made up of chemicals.
What is a chemical change?
A process that alters the arrangement of atoms in a substance.
What is a van der Waals radius?
The radius of an imaginary sphere that can be used to represent the effective size of an atom.
What is a lattice?
A regular array of atoms or ions.
Why did Heinrich Otto Wieland, Germany, win the Nobel Prize for Chemistry?
For his investigations of the constitution of the bile acids and related substances. The bile acids are a set of steroid acids whose synthesis begins in the liver with the production of chloric acid, chenodeoxycholic acid, all of which derive from cholesterol. Wieland isolated and determined the structure of a number of these biochemically significant compounds. During his career he also isolated toxins from poisonous frogs and mushrooms.
What is a neutron?
Neutrons are the other principal component of the nucleus of an atom, along with protons. The neutron is neutral in charge and has a mass roughly similar to that of a proton. Atoms of the same element that contain different numbers of neutrons will generally still have the same behavior as one another in terms of chemical reactivity properties. Both protons and neutrons are, in fact, made up of even smaller particles. But chemistry doesn't usually deal with these even smaller bits.
What is splenda, R?
Splenda, R, is a commercial name for an artificial sweetener, like NutraSweet, R, or Equal, R. Sucralose is the key sweet ingredient in Splenda, are, but unlike natural sugar molecules. Sucralose is not metabolized, so it effectively has 0 calories. It's made from regular table sugar by selectively exchanging 3 O groups for chlorine atoms.
Why do we need to sleep?
Even though this sounds like a pretty straightforward question, the truth is that scientists still don't know the whole answer. There are several theories, including the idea that sleep promotes restorative functions. That sleep promotes development and structural changes in the brain, that we sleep because it helps us to conserve energy. Or simply that it may have been safer for our evolutionary predecessors to remain inactive at night. While there is evidence to support each of these, and other theories, it remains a very difficult question to answer conclusively.
How can I observe layers of immiscible liquids?
Chemistry principles encountered in this experiment: density, miscibility, polarity. Materials you'll need: asterisk. Honey, pancake syrup, liquid dish soap, water, vegetable or cooking oil, rubbing. Alcohol, lamp oil, a tall glass of water or other container, optional. Food coloring to improve visibility, asterisk. Note: for this experiment, it is not necessary to have every material listed. The procedure: pour the densest liquid into the glass first. Note that the liquids above are listed from most dense to least dense. Try to avoid letting the liquid run down the sides of the glass. Gently pour the second liquid on top of the first. One way to pour it a little more slowly is to pour the liquid over another object such as a butter knife or the backside of a spoon. Allow each layer of liquid to settle for at least a few seconds before adding the next liquid. You'll notice that, instead of mixing, the liquids tend to stay in separate layers. The reason for this is that they are immiscible, which means that it is more thermodynamically favorable for the liquids to stay separated in layers and to form an interface than it is for them to mix together. Whether or not two liquids will be miscible is dictated by the details of the entropic and enthalpic factors associated with the mixing or separation of the two liquids in question. This is often directly related to whether or not the compounds have similar polarity. For example, we know water is a very polar substance, while vegetable oil is composed of primarily long, nonpolar hydrocarbon chains. These do not interact favorably with one another, and prefer to stay in separate layers. Continue pouring the third, fourth, etc. liquids on top of each other in order of decreasing density. You can just follow order of your liquids in the list above. As you pour in the successive liquids, they should continue to form separate layers. The densest liquids are affected the most by gravity, so these tend to stay below the less dense liquids. These liquids are not miscible, so they do not mix together to form a single solution. In truth, if you wait long enough, some of these liquids will mix together. But it will take a while. That's it. You should now see a series of separate liquid layers in your container.
Why did Svante August Arrhenius, Sweden, win the Nobel Prize for Chemistry?
For his electrolytic theory of dissociation, Arrhenius began studying as a graduate student at the University of Uppsala how much electricity solutions of various molecules could conduct. He correctly proposed that solution of salts were conductive because the salts formed charged particles, later named ions, even in the absence of an electric current. His advisors weren't very impressed, and he earned a third-class degree in 1884. Arrhenius continued to develop the work, and was soon awarded the Nobel Prize for his insights.
What is homogenized milk?
Homogenized milk is milk that won't separate. Normally cream will separate out from milk, forming a layer at the top of the bottle. This is obviously not ideal, so to prevent this separation from happening, milk is treated with pressure to break up the little clusters of fat into much tinier pieces. These tiny globules of fat don't recombine at an appreciable rate, so the milk remains a single layer throughout its shelf life.
Is our Sun unique?
Aside from the fact that we're spinning around it, not really. It's a yellow dwarf star of average size, 6.960 x 108 m radius, 1.989 x 1030 kilograms, and surface temperature, 5500 to 6000 K.
Why did Arturi Ilmari Virtanen, Finland, win the Nobel Prize for Chemistry?
For his research and inventions in agricultural and nutrition chemistry, especially for his fodder preservation method. Virtanen was quite an interesting chemist, and he was also a farmer. His fodder preservation method made use of hydrochloric and sulfuric acids to slow the processes that normally cause fodder to ferment. He also made several other achievements in the area of nutrition/agricultural chemistry, helping to better provide nutrition to animals raised on farms.
What chemical process leads to the formation of fossils?
There are of course many different types of fossils, but most form through some sort of mineralization process. What's that? In water that has lots of dissolved minerals, after an organism dies. Those minerals can slowly deposit in the tiniest of spaces within that organism, even within cell walls. Well-preserved fossils require the organism to be covered with sediment quickly after death. Like on the bottom of a lake, so that the body doesn't decay before the slower mineralization process can take place.
Why are metals attracted to metal? Or why do magnets stick to your fridge?
Ferromagnets generate their own magnetic field. This means that other paramagnetic substances will be attracted to ferromagnets. Just like they are attracted to magnetic fields. Most metals are paramagnetic, so magnets stick to your metal fridge. But your fridge itself is not magnetic.
Is it possible to see a single molecule?
With some of the largest single molecules, like polymers, they can actually be seen by the naked eye or through a microscope. Most molecules, however, are so small that a single isolated molecule cannot be seen with even the best microscopes. There is a physical limitation that prevents their observation with light, which has to do with the size of small molecules, lengths of ca. 0.1 to 1 nanometer, being significantly smaller than the wavelengths of visible light, 400 to 700 nanometers. Other techniques based on diffracting electrons off of molecules, measuring the force molecules exert against a very small metal tip. And other methods have been developed to image small molecules. But it's impossible to see most small molecules with light in the way that we conventionally see things.
Why did Jaroslav Hayrovsky, Czechoslovakia, win the Nobel Prize for Chemistry?
For his discovery and development of the polarographic methods of analysis. Hayrovsky was an innovator in the field of chemical analysis, developing a polarographic method of analysis capable of analyzing the presence of virtually any species dissolved in water, and could determine the relative percentages of the species present. He did this using a method that we won't get into here, but it relied on a fairly simple procedure involving the application of a current and measuring drops of mercury.
How does an atomic bomb work?
Atomic bombs, A-bombs, are based on nuclear chain reactions that occur very rapidly, causing a huge release of energy in a very short amount of time. In early designs, two pieces of uranium would be fired at one another in the core of the bomb, initiating the fission chain reaction responsible for the explosion of the bomb. As the bomb starts to detonate, the core of the bomb expands. and it is necessary that pressure be applied against the expanding core while the fission process takes place. Within a fraction of a second after detonation, the explosion takes place. These are the type of bombs that were used at Hiroshima and Nagasaki in World War II and are the only nuclear weapons that have been used in war to this day.
Why did Robert Burns Woodward, United States, win the Nobel Prize for Chemistry?
For his outstanding achievements in the art of organic synthesis. Woodward made tremendous research achievements in a broad range of areas in the field of organic synthesis. He established the structures of aureomycin and terramycin, which are antibiotics, and made possible new synthetic work in this area. He also synthesized quinine, which was considered a great challenge and was used to fight malaria. And later synthesized cholesterol and cortisone. The list of synthetic achievements goes on and on. And he truly ranks among the most successful synthetic chemists ever to have graced the field. In addition to these synthetic achievements, Woodward also established the structures of a large number of important compounds.
What is an enzyme?
A protein-based molecule that acts as a catalyst.
What is a meniscus, and how should it be read?
A meniscus is the curve in the surface of a liquid contained within a usually narrow container, such as a graduated cylinder. Take a look at the graphic to the right. The numerical value we read for the volume of liquid contained in this cylinder will depend on what part of the meniscus we choose to look at. Graduated cylinders and other containers are calibrated such that we should get the most accurate reading by looking at the center of the meniscus from eye level. If we don't look at the meniscus from eye level, something called parallax error will occur that will distort our perception of level of the meniscus.
What is a reductant?
A reducing agent, or a species that gives electrons to another species.
What is an organic compound?
Any molecule, or compound, that contains carbon atoms is referred to as organic. The usage of this term is a bit arbitrary, though, as some forms of carbon, like graphite and diamond, and carbon-containing ions, like formate and carbonate, are not thought of by chemists as organic molecules.
What is hapticity?
Number of contiguous atoms coordinated to a central atom.
Why have chemists devoted so much focus to the chemistry of carbon?
Because it's everywhere. Carbon is the fourth most abundant element in the universe, 15th on our planet. And the building blocks of life, DNA, amino acids, all contain many carbon atoms. Many biologically active molecules and medicines rely on carbon to define their overall shape.
What gives some people such a high alcohol tolerance?
From the biochemical point of view, the amount of the enzyme alcohol dehydrogenase in a person's body determines how rapidly their body can process alcohol. People with more alcohol dehydrogenase can convert ethanol, which is what gets you drunk, to acetaldehyde faster. A person's body size also is important, though. Bigger people have more mass to spread the alcohol around, so their blood concentration of ethanol doesn't rise as fast as that of smaller people.
What is a chemical bond?
Sharing of electrons between two or more atoms.
How does a pregnancy test work?
A pregnancy test works by detecting a hormone called human chorionic gonadotropin, HCG, that is produced in the placenta after fertilization. The HCG hormone will be present in the blood and urine about a week after conception. And it's detected by an indicator molecule on the pregnancy test that changes color upon binding to HCG. The tests you can buy in the store are actually pretty similar to those used at a doctor's office. Both are based on HCG binding to a chemical indicator that changes color. The main difference is that the doctor's office has technicians who are more familiar with using the tests. So there's less chance of a mistake with using or interpreting the test.
How is rainfall affected by pollutants in the atmosphere?
Air pollution can affect local and global weather patterns, including the amount and frequency of rainfall. At low concentrations, particles floating around in the atmosphere may help clouds and thunderstorms to develop. But as their concentration increases, these same particles can inhibit the formation of clouds that give rise to rainfall and thunderstorms. This topic is also relevant to issues surrounding climate change. Since clouds are understood to generally have a cooling effect on the climate because they reflect incoming sunlight.
Why did Venkatraman Ramakrishnan, United States, Thomas A. Stites, United States, and Ada Ionath, Israel, win the Nobel Prize for Chemistry?
For studies of the structure and function of the ribosome. Ramakrishnan, Stites, and Ionath are responsible for uncovering the structure and function of the ribosome at the atomic level, and it is for that work that they share this Nobel Prize. They used X-ray crystallography to individually map each atom that makes up the ribosome. It consists of hundreds of thousands of atoms. The ribosome plays a crucial role in each cell, as it is responsible for the synthesis of proteins. On the basis of their results, these researchers have also developed models showing how antibiotics bind to the ribosome.
How can I inflate a balloon using yeast?
Chemistry principles encountered in this experiment: biochemical/enzymatic reactions, gases, materials you'll need. Yeast, 5 to 10 grams powder yeast, a small plastic bottle, preferably about 16 ounces. Or smaller, 1 teaspoon of sugar, a balloon, warm water. The procedure: add enough water to a small plastic bottle to fill it up about 1 inch. The water needs to be warm for the yeast to do its job. Add about 5 to 10 grams of yeast, 1 small packet will do, to the bottle, and mix it around well. Yeast is made of fungal microorganisms that will become active when placed in the warm water. Add a teaspoon of sugar to the water. The sugar serves as the food for the yeast microorganisms. They are going to consume it and produce carbon dioxide gas as a byproduct. Wrap the balloon around the open end of the bottle. You may wish to use tape or a tight rubber band to prevent gas from escaping. Let the yeast do their job for about 20 minutes. And the yeast should soon produce enough carbon dioxide gas to start filling up the balloon. This is the same thing that happens when you use yeast to bake bread. The little holes you see in the bread are from the yeast releasing carbon dioxide as it rises. Try repeating the experiment using different amounts of water, sugar, and yeast, and observe the rate at which the balloon inflates. One hypothesis you might test is whether the concentration of sugar in the water affects the rate of carbon dioxide product. You might also try varying the amount of yeast you use. Pay attention to both the rate at which the balloon inflates, and the final volume of gas it reaches.
What does that mean?
Average kinetic energy is a precise way of saying how fast something is moving. In this case, on a molecular level. The faster molecules vibrate. The hotter they feel because heat is being transferred from the object to your hands.
What is a nonmetal?
An element that does not possess the properties of a metal.
What do the terms miscible and immiscible mean?
Miscible and immiscible are terms that refer to whether two liquids that come into contact will mix to form a single uniform solution. Two miscible liquids are able to mix together to form a uniform solution, while two immiscible liquids will separate into two layers. If the liquids are immiscible, the liquid with the lower density will stay on top and the liquid with the higher density will be on the bottom.
What makes string cheese stringy?
In the United States, string cheese is usually mozzarella, sometimes with some cheddar thrown in. The production process takes melted cheese and stretches and folds it in a single direction. This stretching aligns the proteins in the cheese, making it possible to peel off long strings of it.
Why does elevation matter for cooking times when I'm boiling water?
If you're boiling water in Denver, the temperature of that water will be about 5 degrees Celsius lower than if you were boiling water in Miami. Because Denver is about a mile above sea level, there is less atmosphere pushing down on that pot of water than there would be at sea level. The decreased temperature that water boils at means that you'll need to increase cooking times the higher up you go.
What is the greenhouse effect, and how does it affect Earth's temperature?
The greenhouse effect involves the thermal radiation from the Earth's surface being absorbed and re-emitted by gases in the atmosphere, termed greenhouse gases. The re-emitted radiation, much of which is in the infrared region of the spectrum, is sent out in all directions, meaning that some of the energy is sent back down toward the lower atmosphere and the Earth's surface. This results in an overall increase in the surface temperature due to the presence of the greenhouse gases. It should be noted that a certain amount of this greenhouse effect is entirely natural, but the effect can be increased when additional greenhouse gases are introduced into the atmosphere as a result of human activity. As a side note, the name for this effect arises from the fact that greenhouses allow solar radiation to pass through glass and remain inside. In fact, a greenhouse operates on a different principle. The presence of the glass walls in a greenhouse prevent heat from being lost due to convection currents.
What is voltage?
The difference in electrical potential between two locations.
How did scientists determine that atoms consist of electrons, neutrons, and protons?
Originally, atoms were thought to be the smallest unit of matter. But in the late 19th century, experiments allowed scientists to finally probe inside atoms. Some of these first experiments were carried out by the British physicist J. Thomson, who discovered the electron. He noticed that the rays, actually rays of electrons, though he didn't know it at the time, were deflected by electrically charged plates, and concluded that these rays must consist of charged particles that were much smaller than atoms themselves. Thomson's first graduate student, Ernest Rutherford, continued to investigate the nature of the atom. In the early 20th century, Rutherford carried out a now famous experiment, in which radioactive particles were shot through extremely thin gold foil. While some bounced off of the nuclei in different directions, most of the particles actually passed through the foil undeflected. Rutherford interpreted this as an indication that the atoms making up the foil must consist of mostly empty space. Over his career, he developed the picture of the atom as a positively charged center surrounded by electrons. And he also proposed that there must be neutral particles, neutrons, to explain the different isotopes of a given element.
What is testosterone?
Testosterone is a steroid hormone molecule that is found in human males and females as well as many other species. In humans, testosterone serves as the primary male sex hormone, and it plays a crucial role in the development of the male reproductive system. In males it is secreted from the testicles, and in females it is secreted from within the ovaries. Males use significantly more testosterone than females, and for this reason males produce testosterone at about 20 times the rate of females. Oddly enough, males are also less sensitive to testosterone than females.
What is an unsaturated compound?
A molecule with at least one pi bond or ring structure.
What is cellulose?
Cellulose, see diagram below, is linear polysaccharide poly for many and saccharide means sugar. So cellulose is a chain of sugar molecules. It's an amazing molecule, it's the most abundant organic compound on the planet because it is the main component in plant cell walls. Cellulose is highly crystalline because of the way the sugar molecules are connected and because of the fact that it's made up of a single enantiomer of glucose. Like polypropylene, highly crystalline polymers like cellulose are very strong enough to make trees stand up straight.
What is the Mohs scale of hardness?
Substances, mostly minerals, are ranked by their ability to scratch one another. If one mineral can scratch another mineral, it gets a higher ranking on the Mohs hardness scale. When Friedrich Mohs devised the scale, diamond was the hardness substance known. So it was given a value of 10. Talc, also known as talcum powder, being very soft, has a hardest rating of only 1. Mohs devised this scale to help sort out the private rock collection of an Austrian banker, and later the Archduke's Museum collection. There are more quantitatively accurate measurements of hardness available today. But the simplicity of the Mohs scale keeps it relevant and practical.
Why did Peter Agar, United States, win the Nobel Prize for Chemistry?
For discoveries concerning channels in cell membranes, for the discovery of water channels, and Roderick MacKinnon. United States. For discoveries concerning channels in cell membranes, for structural and mechanistic studies of ion channels. Agar received half of this prize for isolating a membrane protein that serves as a water channel in cells. For roughly 200 years it had been postulated that cells contained channels capable of moving water in and out of cells. But Agar was the first to finally discover a concrete example of a water channel. MacKinnon received the other half of this prize for his work on ion channels, and in particular for his characterization of the spatial structure of a potassium channel. For the first time, this allowed chemists to see how potassium ions flowed in and out of cells.
What is an alpha particle?
A particle consisting of 2 neutrons and 2 protons, i.e., a helium nucleus.
How do the atomic radii of atoms change across the table?
The atomic radii of atoms generally decrease going from left to right across a period, and increase going top to bottom down a group. See graphic on next page. The increase in atomic radius going down a group is fairly straightforward to understand. Additional shells of electrons are added and they must surround the inner shells, resulting in an increased atomic radius. though the number of protons in the nucleus increases going down a group. The inner shells of electrons serve to shield the valence shell from the attractive force of the nucleus, resulting in an overall increase in atomic radius. Moving to the right across a period, the number of protons increases, increasing the attractive force on electrons in the valence shell. Within a period, additional electrons go into the same valence shell, and an increasing attractive pull from the nucleus results in a more contracted valence shell, resulting in a smaller atomic radius. The situation is complicated by the rightmost group, known as the noble gases. But the atomic radius of these elements is typically not important as they are rarely involved in chemical bonds to other atoms.
What is the antibonding orbital?
Orbitals in which the component atomic orbitals are out of phase, leading to repulsion or destabilization.
What is a noble gas?
Group 18 of the periodic table. Characterized by their general inertness due to their complete valence shell of electrons.
What wavelengths of light from the Sun reach Earth's surface?
The light from the Sun reaching the Earth's surface spans a range of wavelengths between roughly 300 to 500 nanometers, with a few gaps in between where atmospheric water and carbon dioxide absorb radiation. The range of wavelengths spanned by light in the Earth's upper atmosphere is slightly broader, demonstrating that gases present in the atmosphere absorb a notable amount of light before they reach the Earth's surface.
What is the rate determining step?
The slowest step in a multiple-step chemical process. Usually the step with the highest energy transition state.
What's special or different about organic foods?
While the precise definition is still changing, everyone agrees that organic foods are those grown without the use of pesticides or synthetic fertilizers. The organic label also frequently excludes the use of irradiation and genetically modified fruits and vegetables. Whether the food tastes better, or is healthier to eat, is for you, or at least someone other than us, to say.
What is precipitation?
The process through which a substance comes out of solution, usually due to insolubility.
What is scientific notation?
Scientific notation is a commonly used method for expressing large numbers. The number is written as a product of a decimal number and a power of 10. See the following question for an example of where this is useful.
Where do we get uranium?
Uranium is mined largely in Kazakhstan, Australia, and Canada, although the U.S., South Africa, Namibia, Niger, Brazil, and Russia are also significant producers. Uranium is mined using a leaching process that allows it to be dissolved from ore deep in the mine and then pumped to the surface, where it is extracted and concentrated for use.
What is kinetic energy?
Energy of an object due to its motion.
What is a mixture?
A system composed of two or more different substances.
What is graphene?
In one sense, graphene is an unrolled carbon nanotube, or a flattened buckyball. Graphene is a material made of carbon atoms arranged in a hexagonal, honeycomb lattice. It is similar to graphite, except that it is only one sheet of atoms thick. A square meter of graphene weighs less than a milligram. Since its discovery, graphene has garnered significant attention for its electronic, thermal, optical, mechanical, and other properties. There is currently a huge amount of research into the properties and applications of graphene. And one Nobel Prize has been awarded, the 2010 Prize in Physics, for research into its properties.
Why does the fifth principle of green chemistry seek to avoid the use of auxiliaries?
Ideally, one would minimize the use of auxiliary substances used in a chemical synthesis, since doing so would generally be expected to reduce the amount of waste produced, thus minimizing the potential for environmental hazards.
Why did Peter Josephus Wilhelmus Debye, the Netherlands, win the Nobel Prize for Chemistry?
For analyzing molecular structure through his investigations on dipole moments and the diffraction of X-rays and electrons in gases. Debye developed the theory of how electric fields affect molecules, and figured out a method to determine their dipole moments by measuring how their insulating properties and density vary with temperature. He also measured interferences of X-rays and electrons with molecules in the gas phase. Along with other investigations into molecular structure, to determine the chemical structures of molecules. This work provided some of the first detailed structural characterization that allowed chemists to definitively determine the different structures of isomers, compounds of the same chemical composition but different geometrical arrangements of the atoms.
How do your muscles work?
Muscles allow you to exercise and move heavy objects and are necessary for basic activities like breathing, pumping blood, and pretty much anything else you do. On a molecular level, muscles work based on the binding, movement, and rebinding of molecules called actin and myosin. This process involves the hydrolysis of ATP to generate energy. For your muscles to move, myosin first attaches to actin, forming a bridge. At this point, ATP and a phosphate group are attached to the myosin. The myosin bends. This is what actually controls movement, releasing the ATP and phosphate. Then a new ATP molecule binds again, and then the myosin releases the actin. The ATP is then hydrolyzed, putting the myosin back into its original position. At which point the cycle can begin again.
Why did Vincent du Vinod, United States, win the Nobel Prize for Chemistry?
For his work on biochemically important sulfur compounds. Especially for the first synthesis of a polypeptide hormone. Du Vinod was known for bridging the fields of organic and biochemistry by synthesizing biochemically relevant compounds. He synthesized the first polypeptide hormone and developed knowledge of the chemistry surrounding peptides, especially those containing sulfur. In early experiments on the posterior lobe of the brain, du Vinod had noted the high percentage of sulfur present, prompting his career-long investigation into the correlation of sulfur with biological activity.
What is beta particle?
An electron created during nuclear decay reactions.
What is a centrifuge used for in a chemistry lab?
In chemistry labs, centrifuges are typically used for separating suspensions. A suspension is a heterogeneous mixture containing small solid particles suspended in a liquid. The number of applications in biochemistry and biology laboratories probably far outweighs those in pure synthetic chemistry. Centrifuges are often used to separate the contents of homogenized cellular material to isolate the proteins or cellular organelles. Centrifuges have also found applications in controlling the rates of reactions by simple partitioning of reactants. A centrifuge can be used to separate enzymes from their substrates in solution, which can serve to stop or significantly slow a reaction that is already taking place. In other cases, centrifuges have been used to attempt to accelerate reactions by forcing reactants together at the bottom of the centrifuge tube.
Is it true that coffee can help you to sober up faster?
Unfortunately, no. Intoxication is related to the amount of alcohol, ethanol, in your body. And the quantity of alcohol will only decrease as your liver works to metabolize the liquor. Drinking caffeine won't really do much to accelerate your liver function. So coffee cannot, in fact, sober you up. It may help you to stay more awake or alert, but you'll still be intoxicated.
Lipscomb, United States, win the Nobel Prize for Chemistry?
For his studies on the structure of borans illuminating problems of chemical bonding. Boron hydride, borane, complexes participate in many interesting examples of chemical bonding. And this is due to the fact that boron possesses one less electron than carbon to donate to chemical bonds, but still often forms four coordinate complexes. Lipscomb won the prize for his pioneering explorations of the chemistry and bonding in borane complexes using X-ray diffraction and quantum chemical calculations. He reached a level of understanding of borane complexes at which he could predict the properties and reactivity of borane complexes reasonably well. And his work led to deeper insight into the nature of the chemical bond.
What is a significant figure?
A digit in a number that is known with some amount of accuracy.
What is quantum mechanics?
The branch of physics that describes the motion and interactions of subatomic particles.
What is malleable?
A material that can be pressed into shapes or sheets without breaking or cracking.
What is permafrost?
Permafrost is soil that is below the freezing point of water for 2 consecutive years or more. Most of it is located near the north or south poles or at very high altitudes.
How do hydrogen-powered cars get their energy?
Hydrogen-powered cars are based on fuel cells that store hydrogen, or H2 gas, inside a material called a polymer exchange membrane. The fuel cell contains two electrodes, an anode, negative side, and a cathode, positive side. At the anode, the H2 molecules are split into protons and electrons. The protons pass through a polymer exchange membrane, while the electrons are unable to pass through this membrane and thus have to flow in a different direction. This creates a current of electricity by which the car is powered.
What is crystallization?
Formation of crystals from a solution of the compound, often used as a purification technique.
Is starch different than cellulose?
Starch is also a polysaccharide, like cellulose, but it is much less crystalline. The major component of starch is amylopectin, which is a highly branched polymer, while cellulose is strictly linear. These branches prevent amylopectin from crystallizing as well as cellulose. Starch is an excellent source of energy, or stored sugar, for plants and animals for these reasons. Because it is less crystalline than cellulose, it is more soluble than cellulose. And the branched structure also means there are more end groups at which enzymes can start chewing the polymer apart.
Florey, United States, win the Nobel Prize for Chemistry?
For his fundamental work, both theoretical and experimental, in the physical chemistry of macromolecules. Florey contributed greatly to polymer science by developing metrics for characterizing polymers and for comparing different polymers against one another. This was a task that had often proved difficult due to the different compositions and conformational arrangements found in different polymers. He made great strides toward putting polymer science on a firm theoretical footing, which was lacking when he entered the field.
How do OLED screens work, and what are they made of?
OLEDs, organic light-emitting diodes, are a class of LEDs, light-emitting diodes, in which an organic material emits light when an electric current is applied. These can be used to create television screens, computer monitors, cellular phone screens, etc. An OLED may employ either small organic molecules or polymers. One advantage of OLED screens is that they do not need a backlight, which allows them to be thin and lightweight and also to display deeper black image levels than backlit screens.
Why did William S. Knowles, United States, and Ryojin Wayuri, Japan, win the Nobel Prize for Chemistry?
For their work on chirally catalyzed hydrogenation reactions and K. Barry Sharpless, United States, for his work on chirally catalyzed oxidation reactions. Half of this prize was awarded to Knowles and Noyori for their work with chiral catalysts that chirally catalyzed hydrogenation reactions, or reactions in which two hydrogen atoms are added. This work found applications in pharmaceutical production soon after its discovery. Sharpless was awarded the other half of this prize for developing chiral catalysts to carry out oxidation reactions, which represent another important class of reactions in organic synthesis.
But what if you were to put a catalyst for gas release into a bottle of Diet Coke, R?
That's exactly what a Mentos, R, does. Dissolved gases need a surface to start forming a bubble, let's just assume that's true, which it is. And the Mentos, R, candy provides a huge amount of surface area because it is a very porous material. So add a Mentos, R, and many more CO2 bubbles can form at the same time, which leads to a sugary eruption.
What makes a bowl microwave safe?
Unfortunately, the only definition is an empirical one. Containers that don't get hot in the microwave are microwave safe. Remember, or go look it up now in the world around us, that microwaves heat food by causing molecules with dipole moments to tumble back and forth. If the container has any such molecules, it'll get hot in the microwave. If any water has leached into your ceramic mug, it'll get hot in the microwave, regardless of whether the container gets hot on its own. The food you are heating up will get hot and transfer that heat to the bowl. So be careful when taking hot things out of the microwave.
What is the Avogadro's number?
The number of particles in 1 mole, 6.022 x 1023.
What is the composition of air?
Earth's atmosphere is made up of 78% nitrogen, N2, and 21% oxygen, O2, if you ignore water vapor, which shifts too much to include in averages for the entire planet. The last 1% is made up of mostly argon, Ar, followed by carbon dioxide, CO2, and other trace gases.
What is a lipid bilayer?
Lipids are important in cells as they form bilayers that protect the cells and hold them together. In a lipid bilayer, the nonpolar tails gather on the inside of the bilayer, allowing the polar ends of each lipid molecule to interact favorably with the polar, aqueous environment of the cell and its surroundings. Within a bilayer, the lipid molecules can still slide around, and can even flip from one side of the bilayer to the other. Lipid bilayers generally don't allow molecules or ions to pass readily, allowing for the buildup of concentration gradients between the cell and its surroundings. Lipid bilayers are also found elsewhere, for example. They can also form separate compartments within cells.
Why are more substituted carbocations more stable?
Great question. First, let's review what led you to this question. In the unimolecular substitution reaction we looked at, an intermediate tert-butyl cation was formed, but in the bimolecular substitution reaction, the cation, this time a methyl cation, CH3, was too high in energy. In this case the hydroxide ion directly displaced the chloride ion. Generally, the more highly substituted a carbocation is, the more stable it is. There are a number of ways to explain why this is true. The first is that carbon substituents are more electron donating than hydrogen atoms. Electrons on neighboring carbon atoms can help stabilize the cationic center. Simply put, the order of carbocation stability matches the number of carbons bonded to the cationic carbon, with higher numbers leading to greater stability.